I guess to start by saying welcome to today's BioPhorum webinar. This is the Accelerating Development Control Strategies for Success webinar. And behind the scenes from a BioPhorum point of view, I would like to thank Jonathan for first of all setting up the webinar and allowing us all here. And secondly, my colleague Sunil for supporting in the background. So if you do have any queries or you've got any problems, please message in the chat and Sunil's here to help as well. As I said, there's already many people on the call. We've actually had over 300 registered for today. So what we've done to make sure that you can hear us and that you've got good audio quality, we've actually automatically muted you, just to let you know. As we go through the presentation, I'd like to invite you to ask any questions that you have. Just pop them in the chat function at any time. That's fine. And what we'll do is we'll come to them at the end, because whilst you'll see two of our presenters today who are from the authoring team, actually most of our authors and most of our contributors are online and available behind the scenes as well. And they'll be able to jump on and answer the questions in that Q&A session too. And a final note before I begin is you've probably seen that the call is being recorded as well. And that's really so we can make it available to people that weren't able to join today. Indeed, yourself, you might want to rewatch this. So we'll make this available to you within the next few days too. So I guess it's worth starting with introducing myself. So I'm Emma Mason. I'm one of our global change facilitators here in the BioPhorum Development Group. And my role really involves, I guess, coordinating and integrating 4 critical work streams within our programme. So that's applied automation, cell line development, CMC speed to market, and in silico modelling as well. And I guess what that involves, because the role doesn't necessarily speak for itself, I suppose, is collaborating with thought leaders, subject matter experts, really looking to address those industry challenges through connecting people from different companies, different teams from all over the globe. And as you can expect, that comes with challenges. So things like time zones, languages, geographical locations, to ensure that kind of cohesive and effective teamwork, because it's all about working together. And ultimately, the purpose of this is to drive that innovation and drive that acceleration in the biopharma industry. So a really great position to be. The other part of this is if you are on LinkedIn, I would love to connect with you. So my colleague Sunil will very kindly put a link to my page in the chat. Feel free to connect, and I'll do that shortly after this. Now, I'm aware some of you might not have heard of BioPhorum before, or maybe you have, but you don't know how all of the pieces fit together. So I'll start by sharing what we are. And I think we often describe ourselves as this coordinated program of change. We really try to create this environment where biopharma and device industry connect together so that they can collaborate to accelerate, I guess, its rate of progress. And ultimately, it's for the benefit of all. So every single party that's connected within that process end to end. We do this by creating this environment where we can collaborate, this model where we can collaborate, that enables these member companies to meet their peers, to talk about their challenges, and really to figure out how they can make things cheaper, faster, and better along those ways. We quite often work on having a consensus, so this consensus-driven approach almost, and this really encourages debate that leads to this understanding, this creativity, allowing the members to share and learn from each other, as well as figuring out those best practice solutions. As you can see on the top right, though, where we've got that circle of circles, this is designed to show our 12 forums, which we currently have. And each of those really provides its own opportunity for companies to align their interests with one another with similarly committed organisations, so it allows that connection. If you want to hear more about BioPhorum, Sunil will be popping a link in the chat shortly. This is to our YouTube channel. We quite often share a lot of the work that goes on behind the scenes here. And as I say, this is where we'll be sharing the recording of this webinar as well in the next few days. But what I want to do now is really zoom into a forum level. So we'll zoom into that development group bubble on that top right and look at the forum itself. So the development group forum, I mean, it does what it says on the tin a little bit. It's kind of focusing on that development of medicines, looking at that point from the receipt of DNA right the way through to BLA or tech transfer, whichever your kind of perspective is. Our mission is really to accelerate and improve. As you've heard, I've used the word accelerate a lot so far, but it is really what it's all about, getting those products to market faster, so ultimately for the benefit of patients. We create these secure environments, we call them work streams, and this is really where like-minded experts can come together to openly share solutions and best practices, discussing things like new industry trends, technologies, common challenges. But it's really important that whilst we do that, we're still protecting company information, particularly when we're thinking about IP. As you can see on the screen, we've got 39 member companies so far, and actually 15 of those are among the top 20 in the industry, which is excellent. But the others also play their part. They might be smaller, but actually they can be more innovative. So I think one of the things I get really excited about is the diversity of the teams that we have here. We have those giants with the resources to invest, but likewise we have those smaller companies that are able to experiment a little bit more and pivot where things maybe don't work or where there are opportunities, they can act that bit faster. So if you want to hear specifically about the development group and all of the other work streams, all the other things that we're doing here, Sunil will share with you the LinkedIn page, which is probably the best way to hear and see all of that. And do get in touch directly if you want to know more as well. But with that, I'll kind of hone into the reason that you're all here, I guess. And that's really because, as I'm sure you know, by the very nature that you're here, the biopharma industry is rapidly evolving. Traditional CMC or chemistry, manufacturing and control, those development life cycles are being transformed. They're being pushed to meet patients' needs faster. So as I say, acceleration is huge. But whilst doing that, we still need to ensure that product quality, particularly when you're looking at accelerated programs. And so within our CMC Speed to Market workstream, a smaller group of people actually came together to look at how they could address that challenge, particularly tasked with developing a comprehensive framework for internal control strategies. And they've published the paper recently, just the tail end of last year, the guidelines to aid control strategy definition for accelerated programs. So that's what this webinar is all about. And what I'll do is hand over to our speakers, so Dan and Julie, give them a chance to introduce themselves and also take control of the screen and yeah, share everything they've got to share. Dan, Julie, do you want to grab the screen?
I think I can do that, right?
Yep, just take control.
Oh, gotcha. I have taken it. Hi, I'm Dan Say. I'm Associate Director of CMC Sciences at AbbVie. I will be the excitement man today. I'm going to go through the early introduction sections, kind of get everyone, hopefully pass on some of the passion I have for this topic to everyone here, so we can have a vibrant discussion later on. Julie, you want to introduce yourself now?
Sure. I'm Julie Beaudet. I am a senior staff scientist at Regeneron Pharmaceuticals in the regulatory sciences space. I will be taking us through a very high-level review of, I think, some 50 pages of impactful and non-impactful parameters that this team identified over a year or so. So you'll hear more from me in the second half of the presentation.
Great. Let me dive right in. So yeah, just overview, kind of just what we went through. I will talk about kind of our purpose and methodology, and then Julie will go through what we discovered through our kind of the process. So yeah, we'll summarize. So realistically, the first thing I wanted to talk about was kind of where we see the work we have done, right? So here is just kind of a really simplified description of how we go from, an early development program to, a commercial program. And the problem being is that, as we all know, that timeline where we have our initial development of our process and go into our commercial process has been continually tightened. We had a common joke that we would cheer ourselves up with at every meeting that, we have no idea what a non-accelerated process is, right? So every time we get something, we're always, how fast can we go? How much faster? And the reasons are good, right? We want to get our products to the patients as quickly as possible. But it does pose a lot of significant challenges for the CMC side as we go in and try to develop robust processes that we feel can support the commercial demand. So really where we focused in our in this initiative is on that first phase of process design, and specifically in how we generate a control strategy to process characterization. So, recognizing that these efficient workflows to get to the characterized and robust process are important, we need to start with a general understanding of how we can do that rapidly. And really, what we wanted to do was look at our standard knowledge that, both in-house and across the whole industry on some of the unit operations that are used for a standard MAB process. So that's where we kind of focused all of our work. We were really looking at how can we take our years of experience and scientific understanding and leverage that to accelerate this generation of the control strategy through process understanding. And how can we take a risk-based approach that really aligns with ICH principles, and go, save the work that we want to do or we have time to do for the most important aspects of the process. So that's really what the team set out to do. And, I won't say how long it took, but we worked very well together for a very long time. And that's what we're going to talk about today. So maybe move forward. So, so I talked a lot about this, right? So we're now looking, as we do drug substance and drug products manufacturing for standard MAB, how can we inform that process characterization activities, right? How can we, again, focus the work that is available to do due to resource and time to those that are most important? And so really taking a step back, looking at first principles where we could, but also just understanding of how, years of platform experience across the industry and across companies to really leverage that experience to minimize those work packages. And that's kind of, these are the key principles we kind of went through there, right? To streamlining to meet those accelerated timelines, and to derive recommendations that are really would really help to enable teams coming in that have an accelerated timeline to focus their energies where they need to be. And again, it's really to deliver to the needs of the patients in kind of the modern healthcare environment, right, where there's a lot of need. We're trying to deliver really robust packages in that short period of time. So, okay, so how do we go about doing this? Initially, we started off with a survey, right? We polled the member companies are part of this work stream, and we got responses to gather this prior knowledge of what companies have previously rated as important parameters in unit operations that should be characterized in process characterization activities to drive these robust control strategies. So one thing that is, I think we could all agree on is there's some parameters that will always be important, right? So pH in a production bioreactor. Everyone is going to say, yeah, you should study pH, right? But what we wanted to really do is try to get consensus and oversight on those parameters that might not be the ones that everyone's going to study, the ones that no one's going to study because in accelerated time you just don't have time maybe to study some parameters in the seed train, right? Or for cell culture, where it's well understood and you have a lot of controls that might help not have the focus there. It's those middle parameters that we really wanted to focus on and provide some experience and information for, and just general overview of what companies have done in the past to help teams as they go through this activity. So we got a lot of responses. We used a simple majority to start classifying parameters, and we really just classified them as, yes, we think these could impact a product, or no, we don't think these could impact. And then that in between, like, we're not sure, but we're going to try to figure out our best case. We did this to, we talked about CQAs, but it really, we wanted to focus on CQAs, of course, but we didn't want to get tied up with, level of impact or, definition of critical versus non-critical. It was really more scientifically, can we just say, will there be an impact from this parameter on a CQA that we think it's relevant to study? This is, again, since it's consensus, we didn't focus on any specific CQA, discreetly for any, we consider a panel of CQAs, common CQAs. The caveat would always be, you have to know your molecule, right? As you go into this activity. It's a standard mAb, but if there's specific liabilities or something, they have to be part of the consideration. So we talked about this a lot, but we agreed that with the common CQAs, how can we do this assessment without getting bogged down and kind of talking about specific molecular liabilities that might have a broader specific indications for a specific product. We also included process performance indicators where we could, like titer, yields. We felt certain parameters maybe didn't impact CQAs, but they would have a big impact that teams would want to know about for process performance indicators. That's something we also considered in this review. Inherent whenever you talk about impact with a parameter to an attribute is the idea of a range. We had a lot of discussions around this, so we wanted to include ranges because kind of talking about an impact without a range is something that is kind of pointless. So we do include typical ranges, right, to where we based our assessment off of. So just to give us a standard ground to work on, they're not operational ranges or anything. The companies were very clear that just wanted to provide a range that they thought was the typical range they might want to explore in a person's characterization. They're not, limits of the ranges that might have equipment tolerances or things like that. I think that's what I wanted to cover. Hopefully, I know I went fast. I talk very fast. I talk very excitedly because I'm very into this topic. But hopefully, just gets the stage set. And now Julie will go through kind of some of the data we have. And I'm hoping to lead into a really robust discussion at the end as the Q&A session. So with that, I will hand it over to Julie. I think Julie, thank you.
Thanks for the hype, Dan. I'm going to now take us through the major sections of the show. So Chinese hamster ovary, cell culture, manufacturing process from vial thaw to final drug product, as well as we'll touch on analytics for a monoclonal antibody. So as Dan mentioned, this is a consensus-based evaluation of all of the hundreds of parameters that were put forward for review. So this is not representative of any one company's approach, nor is it the final end-all be-all of where these parameters fall, as Dan already covered. We'll start out with the cell culture controls right at the beginning of the process to help simplify the way we looked at this, particularly from a facility ability point of view, which we did not consider in scope. So facility specific parameters will not be listed throughout this discussion. We broke it down into vial thaw and inoculum expansion as the first part. Sometimes this is also called cell culture expansion, and then the production bioreactor, the big guy. So in the vial expansion part of the assessment, we identified 2 impactful parameters out of the 20 assessed with a little star next to them because they were considered very highly needed forward processing parameters. These were the initial VCD, so viable cell density, and the temperature of the cell expansions. These were really related to the process performance indicators, but were rated as such that we wanted to call them out here as potentially impactful parameters. In the production bioreactor, we had 10 impactful parameters, and this increase is due to the potential impact on CQAs and process performance indicators, since this is where the majority of our products are produced in the production bioreactor. The consensus at this stage is that the cell expansion stages are mostly considered for impact to cell growth, but at that point, we're not really making our product. So while cell health is something that's important for making high quality product, it doesn't directly impact that product at each stage. And then in the production bioreactor, we have multiple impactful parameters that we are recommending to study due to the formation of the product of interest. I do see a question about if there will be a white paper related to this topic. Yes, there is. It's actually available on the BioPhorum website already. And I know there's a link in these slides, and maybe one of our BioPhorum wizards can put the link in the chat as well. Next, we looked at purification controls. This actually is an amalgamation of some 10 unit operations related to purification. And here we identified 35 impactful parameters shown in the bar graph as the dark teal at the bottom. The light teal represents the non-impactful parameters that were identified, giving us a total number of parameters. These unit operations go from harvest and continue on through formulation steps. We created an umbrella characteristic for those in between. So we did have such commonly called out unit operations is viral inactivation, as well as ultrafiltration, diafiltration, or UFDF. However, when we started talking about specific columns and resins and conditions there, we had multiple meetings of lively conversation before we settled down on a capture chromatography. Something that is commonly used is protein A affinity chromatography in this space, as well as polished columns that could be either flow through or bind in elute. Here, our consensus was that the process-specific performance parameters will likely need to be characterized, especially since we're looking to make that pure product for our patients. And then the determination of impurity clearance can be used to justify control of process and reduce testing at later stages. This is where we start to see more analytic testing, often outlined through International Conference of Harmonization and local regulatory guidance on things like bioburden and endotoxin testing. Drug product manufacturing brought us many more lively conversations. This we broke down from the thaw of the incoming bulk product all the way through packaging. In this 9 unit operations that were reviewed, we identified 36 impactful parameters. I do want to note that we did exclude pre-filled syringes and assembled devices from this analysis. So it is focused on vial drug product. We did include lyophilization as one of the unit operations, with the caveat of if you're considering liquid drug product, you could skip over that section. Here, the impactful parameters are largely related to microbial control, product stability, and the appropriate fill. And then later, we're looking at the influence of container closure system and presentation. This particular unit operation as a whole had a lot of discussion around the facility-specific adaptations that are needed. That, as I mentioned earlier in the presentation, that is out of scope of these parameter studies. That being said, there are a number of specific studies that will likely need to be conducted, particularly around dilution and lyophilization, if those apply to your drug product. Analytical control is the topic that is most near and dear to my heart, and we found that we probably could have written a paper on this on our own. Luckily, our colleagues in the development group's regulatory subteam were already working on one for specifications and how one could harmonize approaches with setting specifications. Knowing this, we decided to scope this down to some general recommendations, including in-process testing, release specifications, as well as stability specifications, and extended characterization. Likely in accelerated development, approaches are going to be reduced because there will just be less time to generate the data sets. Knowing this, we came up with a panel of what we thought were the most critical tests to evaluate in process, in addition to the microbial control samples that are dictated through guidance. We've also summarized some approaches for setting specifications, whether by a statistical approach or prior knowledge. And that is further discussed in the companion paper, Harmonized Approaches for Setting and Justifying Commercial Specifications of Biological Drugs, which is also available on the BioPhorum publication hub. Our consensus from the analytical control is that IPC, or in-process testing of critical quality attributes, should be based off of a risk assessment and prior knowledge. Reduced in-process testing scope in later phases with adequate data is an ultimate goal for these accelerated programs, and that you should definitely take a look at the specification paper. And I'm not just saying that because I'm also on that team. In summary, we've covered a lot of information very quickly. To reiterate, the acceleration of program development to meet patient need is an ongoing fact of life in the biopharmaceutical world, leading to the streamlining of process characterization and control strategy definition. We approach this task through consensus-based review of core process parameters with the high level breakdown that we've covered today, and analytics or measuring process performance and quality, product quality are very important, but largely out of scope of the parameters that we've reviewed. This group has identified these impactful and non-impactful parameters based off of their collective experience. So product-specific knowledge for both the product and the process in the facility that it's being executed in are still a critical part of control strategy definition. These guidelines are meant to be a baseline for establishing meaningful controls. So hopefully it will help you streamline some of your decisions in an accelerated environment. And we have a link to the published paper that was effective and out there November of 2024.
Okay, well, look, that wraps things up beautifully, I think. We've kind of come to the end of our time here. So I think I'd just like to pause and say thank you so much, first of all, to everyone for coming along, because you're the reason that we're here. Secondly, thank you so much to Julie and to Daniel for presenting today. I think you did a wonderful job in conveying so much information. I know so much detail is there behind the scenes, but you managed to put it really succinctly. Thanks to the rest of the team for chipping in with the Q&A. And then likewise, thank you to everyone else that contributed, as I said before. But for now, I will close this webinar and wish you a great rest of your day.